I would like to thank the organizers of the 13th anniversary World DNA Day 2025 for inviting me to give a speech, Rejection of the mRNA Therapies by the Fundamental Theory of Wave Optics in Chromosomes. I'm the author of the Fundamental Theory of Wave Optics in Chromosomes, 1985, and the founder and president of the Wave Genome LLC biotechnological company, founded in 2010. The most urgent task of science at the moment is to prevent the introduction of mRNA drugs, including the anti-cancer mRNA drugs, namely, to reveal the true meaning of mRNA drugs as biological weapons aimed at destroying the human genome of the recipients through gene modification that causes cancer, sterilization, and prion disease. This moment is a turning point in the development of mankind. Gene modification, or turning people into genetically modified organisms that are sterile, is reaching a new level with the use of artificial intelligence. I will present the prospects for anti-cancer and anti-prion therapy based upon my fundamental theory of wave crystals and the wave optics of chromosomes, 1985, and laser bioholography technology. In particular, I will present a supplement consisting of the electret resveratrol and the semiconductor organic bivalent selenium, which together create a semblance of a scalar wave capacitor with the processing of this supplement by a laser and the transferal of the client's individual biohologram onto this supplement. At the inauguration of American President Donald Trump on January 20th, 2025, the Oracle co-founder Larry Ellison announced the launch of the Stargate program as a key project of the Trump administration. Stargate's activities, according to Mr. Ellison, will consist of testing the blood of each citizen daily by the artificial intelligence for the presence of cancer cells. And if cancer cells are detected, the artificial intelligence will instantly produce an individual anti-cancer mRNA vaccine for this person. Anti-COVID mRNA vaccines have led to the emergence of such a phenomenon as turbocancer. Turbocancer is a massive and rapidly developing cancer in all population groups, including children and athletes. World Health Organization predicted in early 2024 that by 2050, the number of cancer cases in the world will increase by 77%. That is why the development of anti-cancer drugs and cancer treatments is the most important task of today for medicine and biology. Another question is whether we can allow mRNA drugs, which cause the phenomenon of mass turbocancer in the first place, to be offered as an anti-cancer drugs. Still another question is whether Big Pharma and the US Food and Drug Administration knew in advance that mRNA drugs would lead to the mass turbocancer. The answer is yes. Evidence that the FDA knew that mRNA drugs would cause mass turbocancer is contained in the FDA-owned documents from 2007, as indicated by Dr. Joseph Ladapa, Florida State Surgeon General, in a statement dated December 6, 2023, published on the official State of Florida website. Even more ominous assessment of the mRNA drugs comes from Dr. J. Bart Claassen in his 2021 paper, COVID-19 RNA-based vaccines and the risk of prion disease. Dr. Claassen claims that mRNA drugs cause prion disease. Prion disease causes proteins to lose their crystalline structure and become like sponges. They cannot transmit electrochemical signals, so that the connection between neurons, the connection between the brain and the body is lost, and the person literally goes crazy. Dr. Claassen concludes his article by saying, many have raised the warning that the current epidemic of COVID-19 is actually the result of an bioweapons attack released in part by individuals in the United States government. Dr. Claassen continues, approving a vaccine utilizing novel RNA technology without extensive testing is extremely dangerous. The vaccine could be a bioweapon and even more dangerous than the original infection. In his 2023 paper, Possible Treatments for COVID Vaccine-Induced Prion Disease, Dr. Klassen is even more categorical. Many COVID-19 vaccines are considered bioweapons and are known to have the ability to cause prion disease. Dr. Klassen emphasizes there is little doubt that the COVID vaccines cause prion disease. Both the vaccine mRNA sequence and the vaccine-induced spike protein have the ability to induce prion disease. And further, the current bioweapon attack with COVID-19 infections and, more importantly, COVID vaccines has definitely been linked to Israel's Mossad. The COVID-19 associated spike protein found in the virus and produced by the mRNA vaccines is a race-specific bioweapon having low affinity for the ACE2 receptor variant which predominates in Ashkenazi Jews. The foundations associated with Bill Gates, who is linked to Mossad operative Jeffrey Epstein, and Larry Ellison, who is also linked to Mossad, have funded prion research, says J. Bart Klassen. And more recently, US congressional hearings have taken place to convince the public that the blame for the attack should be placed on everyone but the culprit, Israel's Mossad. Part 1. Why mRNA drugs cause cancer and prion disease and are bioweapons. Any RNA virus, bird flu, influenza A, SARS-CoV-2, Zika, Ebola is simply a broken coherence of the crystalline structure in the wave optics of chromosomes. Therefore, no RNA virus can be isolated from the intracellular fluid. There are as many strains of RNA viruses as there are people. Luc Montagnier made false claim that he had isolated the HIV, human immunodeficiency virus, because HIV cannot be isolated from the intracellular fluid in principle, and because all human RNA viruses are HIVs, including SARS-CoV-2. All human RNA viruses are HIVs because to be infectious to humans, an RNA virus must have nucleotides homologous to human nucleotides. Homologous nucleotides fit into each other like a key into a keyhole. So these extra superfluous nucleotides destroy the precisely calibrated geometry of chromosomes. There must be a precise number of nucleotides in a chromosome, no more, no less. And structural damage to chromosomes is the major cause of disease. When the chromosomal structure is destroyed, the cell divides incorrectly. Structural damage to chromosomes is the main cause of cancer and prion disease. Moreover, structural damage in chromosomes leads to sterilization. The more the chromosomal structure is damaged, the more rapidly the cancer progresses. And sterilization occurs faster, not in the third generation, but already in the second or even in the first. It does not matter whether these superfluous nucleotides are delivered via the DNA fragments or the RNA fragments. So the immune system tries to get rid of these superfluous nucleotides. And since these superfluous nucleotides get inserted into the RNA and DNA of a human, the immune system attacks itself. Herefrom we get all kinds of the autoimmune reactions, such as Cytokine Storm, Antibody-Dependent Enhancement, and Pathogenic Priming. RNA virus-caused autoimmunity means that first, no immunity to the RNA infection is developed, and therefore vaccines against the RNA HIV viruses are impossible in principle. Second, repeated infections with the same or similar RNA virus cause even more serious disease than the initial infection. And third, the level of antibodies does not indicate the level of immune protection against the RNA virus, but on the contrary, the more antibodies a person has, the sicker he is. When the immune system destroys itself, we get AIDS, the Acquired Immunodeficiency Syndrome, and the patient can die from even the slightest infection. That is why Luc Montagnier intentionally or unintentionally misled the world public with his claim that he isolated HIV, based upon his false claim that he isolated HIV, human immunodeficiency virus, it became possible to falsely claim that HIV was transferred to humans from African monkeys, although in nature there is the interspecies barrier. That is, monkeys do not have nucleotides homologous to human nucleotides, and that is why the animal viruses cannot, in principle, be transferred to humans. Therefore, any bird, swine, monkey, or mouse virus transmitted to humans is definitely a product of a military biological laboratory and a binary biological weapon. A binary biological weapon means that two viruses are combined. In the case of SARS-CoV-2, an animal respiratory virus, and a human RNA virus, that is the human immunodeficiency virus, were combined. This was done by Professor Ralph Baric at the University of North Carolina by simultaneously infecting humanoid mice with inserted human genes with two viruses at the same time, the human immunodeficiency virus and an animal respiratory virus. As a result of these experiments by Professor Baric, 4 HIV inserts were made into the surface protein S protein. Such developments of the binary biological weapons are officially called the gain-of-function research and the dual-use research of concern in the USA. Continuing with the same logic, all further mRNA drugs are also the dual-use research of concern or biological weapons, as they cause the structural damage in chromosomes, which leads to cancer, AIDS, sterilization, and prion disease, that is insanity, and even more if mRNA drugs are individualized by the artificial intelligence, as Larry Ellison promised us, then danger will increase exponentially. This is proven by the discovery of the destructive RNA interference effect by Craig Mello and Andrew Fire, for which they received the Nobel Prize in 2006. They took RNA from the C. elegans worm and gave it back to the worm, and the worm died. The worm died because the superfluous nucleotides broke the geometry of a chromosome, that is, caused the structural damage in the chromosome. The effect of the destructive RNA interference proves that first, the introduction of the homologous nucleotides through mRNA drugs is deadly. Second, the genetic therapy with one's own nucleotides through the mRNA drugs is just as dangerous as with the donor's genetic material. And moreover, the genetic therapy with one's own genetic material is more dangerous than with the donor's genetic material, because one's own genetic material is more homologous than the donor's genetic material. Third, President Trump's mRNA anti-cancer program is in fact a program to sterilize the American population and the population of the entire world, with the accompanying mass epidemic of cancer on the way to mass prion disease, the so-called zombie apocalypse. The effect of the destructive RNA interference is in fact the process of gene silencing, which occurs when the homologous nucleotides are inserted into each other like a key into a keyhole. And the more homologous the nucleotides are, the more destructive the effects of the mRNA drugs will be. And the more often the mRNA drugs are administered, the stronger the effect of the destructive RNA interference will be. That is, the so-called mRNA anti-cancer therapy of Larry Ellison will cause cancer instead of curing it. Now, let's consider what the effect of the destructive RNA interference is and why the destruction of the chromosomal structure through the introduction of the mRNA drugs leads to cancer, prion disease, and sterilization. Part 2. Fundamental theory of wave optics in chromosomes as the basis for correcting the structural damage in chromosomes. The world is 93% energy and only 7% particles. Therefore, the HIV condition or the structural damage in chromosomes can be caused and more effectively by the electromagnetic fields such as 5G. And in order to get rid of the HIV condition, that is to correct the structural damage in chromosomes and restore the coherence of the cell liquid crystal media, it is necessary to restore the coherence of the cell's wave crystal media. The dilemma of whether SARS-CoV-2 is an infection or a harmful effect of the electromagnetic radiation distracts us from the main point that the structural damage to chromosomes is in any case the violation of the wave processes in the cell, regardless of whether 5G was used to create the COVID pandemic or not. That is why, in order to cope with cancer, AIDS, sterility, and prion disease, it is necessary to have the fundamental theory of how the wave processes function in the cell and to build biotechnology on the basis of this understanding. In the mid-1980s, I formulated the fundamental theory of wave crystals, and on its basis, the theory of wave optics and chromosomes. The theory of wave crystals is the theory of everything. The essence of the theory is that the being is the infinite and eternal wave crystal, with the focus being one and the same for the chromosome and the galaxy. In wave crystals, information is transmitted by refraction of the signal towards the focus of the wave crystal. The more coherent the wave crystal is, the farther the information is transmitted. The eternity and infinity of the quantum nonlocality implies the absolute coherence of the world wave crystal. And eternity and infinity mean the absolute instantaneity in the transmission of information. The transmission of information not limited by the parameters of time and space. In the world wave crystal, information is transmitted by the zero signal of the zero field. The zero signal is not of the electromagnetic nature and is transmitted instantaneously and remotely according to the principle of the spatial translational symmetry. The world wave crystal is precisely the crystalline structure of the standing scalar wave. The standing scalar wave is the zero wave or the zero signal because the scalar wave annuls any linear signals when the peak of the forward-going wave is nullified by the trough of the same wave when this wave is reflected back upon itself. Each part of the world wave crystal of the scalar wave is also a generator of the form of the standing scalar wave. The wave crystal is a scalar wave diffraction grating with a certain wave optics of refraction towards the focus of the wave crystal of the quantum nonlocality. That is, infinity is not simply extensive across the horizon, but also intensive inwardly. The being exists on the basis of the holographic principle. The holographic principle states that the being is entirely in its every matrix point. Thus, the quantum holography is implied in the very concept of infinity. A holographic signal is a collection of all frequencies, waves, particles, and fields simultaneously. People differ from animals precisely by their ability to recognize the infinity of the being. In August 2012, I formulated the implication of the holographic principle. If the being is entirely in its every matrix point, then each matrix point is not simply different from any other matrix point, but is unique. That is, each matrix point is simultaneously non-local and unique. Matrix points differ from each other by their refraction code. Between two copies of a unique wave matrix, information is transmitted instantaneously and remotely, since in the quantum nonlocality they constitute one and the same systematic whole. The world hologram is a scalar wave diffraction grating modulated by the individual refraction codes. Two unique wave matrices exist parallel to each other and are protected from the arbitrary information transfer between each other. Information exchange between them, including instantaneous and remote, is possible only with an additional structural key, the simultaneous application of a scalar wave diffraction grating with the same refraction code to both of these unique matrices. The holographic principle implies isometry, or the principle of the spatial translational symmetry, from macro to micro dimensions ad infinitum. That is, a chromosome is also a wave crystal formed by a scalar wave diffraction grating and with a certain wave optics of refraction towards the focus of the quantum nonlocality, the zero center of the zero field. The chromosome functions on the basis of holography and the holographic principle, similar to a galaxy. In order to restore the damaged chromosomal structure, we must use devices and drugs that create and modulate the scalar wave diffraction grating in the patient's cell, that is, operate with the wave optics of chromosomes. These devices and drugs must use the laser bioholography because only a laser is capable of creating a coherent signal with a constant frequency to create a scalar wave diffraction grating. The same gene is expressed in dysfunctional individuals by an acrocentric chromosome and in functional individuals by a metacentric chromosome. The ability of chromatids to exchange information and to regenerate and replicate in general depends upon the specific geometry of wave optics in chromosome. If this specific geometry of wave optics is violated, the chromosome loses its ability to regenerate and replicate. The same gene is expressed in humans by a metacentric chromosome, which has the shape of an equilateral cross and is analogous to a well-centered and focused eye. And the same gene is expressed in monkeys by an echocentric chromosome, which is the half of a metacentric chromosome. That is, it demonstrates asymmetry and is analogous to a nearsighted and farsighted eye that has lost focus. Individuals and species with extra-chromocentric chromosomes exhibit mental retardation or some degree of mental degradation, and until now, no one has been able to explain why. I argue that our chromosomes function similarly to our eyes or lenses according to the laws of the geometric optics and holograms. The structural damage in chromosomes is crucial, since a geometric shape literally cannot form a focus or zero center in a hologram in the acrocentric chromosome. For example, the male gender chromosome is acrocentric, and therefore a man is not capable of childbirth. The female gender chromosome is metacentric, and therefore a woman is capable of childbirth. Obviously, the male gender chromosome is the result of the structural destruction of the female metacentric chromosome. This explains why the structural damage in chromosomes inevitably leads to sterility. Only my fundamental theory of chromosomal wave optics can explain why the structural damage in chromosomes leads to sterilization, or in other words, why the mRNA drugs and any other gene modification that destroys the chromosomal structure are causing cancer, AIDS, sterilization, and prion disease. The process of creating a scalar wave crystal is as follows. The scalar wave diffraction grating is scaled in such a way that each segment of the scalar wave itself becomes a scalar wave, from macro to micro dimensions, and vice versa ad infinitum. This creates refraction of the signal, precisely because the scalar wave diffraction grating is the generator of the form. It restores the isometry of the object at each level of refraction. My company, Wave Genome LLC, develops and produces devices and supplements of the laser bioholography based upon my fundamental theory of the wave optics and chromosomes. The devices and supplements invented and produced by my company, Wave Genome LLC, create and modulate the scalar wave diffraction grating in the patient's cells, that is, they operate with the wave optics of chromosomes. Wave Genome LLC uses laser bioholography to create a scalar wave diffraction grating, including red and green lasers. Part 3. Anti-cancer and anti-prion therapy prospects from the bioholographic perspective. Since the world is 93% energy, we can eliminate extra nucleotides that destroy the chromosomal structure only at the wave level through a scalar wave that annuls any external signal when the peak of the forward-going wave is annulled by the trough of the same wave when the wave is reflected back upon itself. That is why we should use the generators of the form that create a scalar wave, including the electrodes, polymer crystal, of the organic and inorganic nature of the plant, insect, animal, etc. origin. But the secret of the correct use of electrodes is their joint use with conductors and semiconductors, since the scalar wave is self-extinguishing, and the combination of the electrode and the semiconductor creates a kind of a capacitor that does not allow the scalar wave to get self-extinguished. A semiconductor or conductor is an antenna for a wide range of electromagnetic signals, including infrared waves. Our blood runs on iron, and our nervous system runs on selenium. That is why the combination of iron and selenium with any kind of electrode will give the best therapeutic result in the case of the structural damage on chromosomes. In a scalar wave capacitor, the standing scalar wave is prolonged long enough to get scaled into scalar wave diffraction grating with the proper refraction wave optics towards the focus of the quantum nonlocality, the zero center of the zero field, one and the same for chromosome and galaxy. The Indians have recently introduced to the world market, after successful clinical trials, a combination of resveratrol and copper as a dietary supplement with a very strong anti-cancer effect. In 2021, my company, Wave Genome LLC, has introduced to the world market a much more potent anti-cancer combination of resveratrol and selenium, organic and bivalent, namely as a scalar wave capacitor. My company, Wave Genome LLC, was the first company in the world to encode electrodes with human wave matrices. Use these human wave matrices for encoding the electrodes as bioholograms, and encode bioholograms into electrodes using laser. Before my company, Wave Genome LLC, other Russian scientists were encoding the electrodes using electromagnetic signals, frequencies. But once again, only lasers are capable of creating a scalar wave, since only lasers create a coherent signal that is a signal with a constant frequency. Thus, Luc Montagnier's experiment of transferring a DNA phantom or the DNA teleportation via the electromagnetic homeopathy is ultimately a profanation. Traditional homeopathy, when the container with the preparation is shaken by hands and the homeopathic preparation is diluted, works much better because each man is a spontaneous generator of the form for creating a scalar wave, a kind of the eternal laser, non-local in time and space. So the rituals of shaking, spinning, dancing in a circular formation with other people in a state of theta rhythms of the brain are an effective way to create a scalar wave diffraction grating. But I repeat, only the laser bioholography can ensure the accuracy of the information transfer. All folk medicine, including Ayurveda, is based on the use of electrodes in the liquid crystal form, the aerosol crystal form, and the solid crystal form. Our ancestors used bombs made from electrodes for thousands of years. That is why each of our large Russian Orthodox churches has the mummified relics of a saint. The body of our saint did not rot, because our saints were eating electrodes, drinking electrodes, inhaling electrodes, anointing themselves with electrodes, and were maintaining their minds in the theta wave brain state, which is a scalar wave, through prayer, rosary counting, and being present inside the church, which is the generator of the form for creating the scalar wave diffraction grating. Our ancestors not only used bombs, but also were wearing the electrodes inside the silver egg-shaped pendants on their solar plexus. Every Russian man before 1917 Bolshevik Revolution was wearing an amulet, ladanka, with frankincense, ladan, hard resin of the incense inside. Ladanka is an effective scalar wave capacitor. Wave Genome LLC produces a quantum leap ladanka, a side generator, encoded with the client's biohologram using a laser. The effectiveness of my company, Wave Genomes LLC electrode-based generators, has been proven by the successful clinical trials at the Bauman Moscow State Technical University, the largest and most important science university in Russia at the MIT level, for the remote transmission of aspirin, the holographic aspirin. Other evidence of the effectiveness of Wave Genomes LLC laser bioholography includes the following. When the site generators by my company Wave Genome LLC were applied to the center of a very black bruise, hematoma, overnight, in the morning the white spots of the completely regenerated bodily tissue were found in the site where the site generators were applied, although the rest of the hematoma remained black. In another case, a site generator was applied to a melanoma for 3 days. After 3 days, the melanoma dried up and fell off. In the third case, a client testified that her long-term atopic dermatitis was cured. The importance of individualizing the electrode-based site generators consist in the following. According to the implication of the holographic principle explicated above, if the being is entirely in its every matrix point, then every matrix point is not simply different from any other matrix point, but is unique. So every wave matrix is both non-local and unique, and we can transmit information remotely and instantaneously between two copies of the wave matrix because they constitute one and the same systematic whole in the quantum nonlocality in which there is no space and time. That is why we can record the client's biohologram with a laser and then encode the electrode with the client's biohologram so that the individualized electrode-based site generator will work as a lens to correct the wave optics of the chromosomes when the client is wearing it on his/her solar plexus. My company, Wave Genome LLC, was the first company in the world to refine the crystalline structure of a dietary supplement, selenium, plus various electrodes such as resveratrol, quercetin, crystal hindi, with the help of a stabilized helium neon laser, and to encode the supplement with the customer's biohologram using the laser.